Thank you. Bridget told you that she was the French lady here. I'm not a French lady, but I'm probably the only federal bureaucrat you're going to hear from today or tomorrow. I work at the National Institute on Aging, where I've been for 16 years. And what I'm going to tell you a little bit about today is aging and lifespan. The National Institutes of Health, in the parlance of capitalization, has a $39 billion capitalization last year. It's a nice piece of change, but we view it as our budget. My institute has $3.2 billion to spend trying to understand human aging. A lot of the organisms that we study in the laboratory are, however, not human. But what we're going to talk about today is what is aging. If you think, now my institute doesn't actually have a definition, so this is just what I make up. So life is getting through the day. There are things called activities of daily living, getting dressed, feeding yourself, going to the bathroom, not necessarily to give a sample for Dr. Kraft's Singularity University, but it's just something that you do. And aging is when you find it more difficult to get through the day. So what do you do to make it easier to get through the day? Are two things. One is you try to slow the rate of aging. If you are experiencing fewer diseases or fewer losses of function at a given age compared to your fellows, not only can they be jealous of you, but you can also feel that you're aging at a slower rate. Some of that is genetic, about 25% of that. 75% of that is what happens during your life course. And you can rejuvenate. So I'm going to focus really on rejuvenation today, but in the broader context, just let me tell you what the general features are of aging. They are decreased resilience. That is, you meet a challenge and it's harder to mount a response. Once you mount a response, it's less effective, and the time between responding to the next similar challenge is longer as you age. There's also, at the other end of the spectrum, for infants, but the problems there are different. When you're an infant, you're acquiring the functions. As you're aging, you're losing the functions that you acquired up to adulthood. Then there's increased frailty. Frailty is actually a clinically useful concept. It's used to tell you how well will the patient survive a surgery, or what's the likelihood of surviving. And there are various metrics for that. And there are about a dozen indices by which you can measure frailty. And there's also increased of chronic disease. This is typical of older people, and the result of having multiple chronic diseases is polypharmacy, which was mentioned earlier. So the keys to slowing aging and promoting rejuvenation are that you remove impediments from the rejuvenation, from the repair or health process. We accumulate a number of things in the course of our lifetime that are deleterious to the function of the human body, and those are impediments to integration of the cells and to good functioning. So, for example, in the heart, there's increased fibrosis. Actually, in all organs, there's increased fibrosis. Fibroblasts and the surrounding matrix, as it's called, don't integrate well with the cardiomyocytes that do the contracting and the pumping. So if you could remove some of that fibrosis, you might have a better functioning heart at an older age. You can also store some missing or factors or things that are at a lower level than when you were a well-functioning adult. And you can then use these to reactivate inherent capacities. We know from some of the research that we support that the older bodies, generally speaking, do have the inherent ability to respond, to have improved resilience, to have improved regeneration capacity. It's inherent, but the circumstances, the things in circulation, things in neighboring cells, are not at a level that will optimize that response. So what can I tell you? I'm going to give you a few examples from biology, aging, and geroscience. I don't have the fancy graphics that everybody else seems to have, but I roll my own. So I'm going to tell you something about various aging tissues and organs from the point of view of if you wanted to do something about aging, which by one estimate is a $137 trillion industry. Because everybody ages, there's 7 billion of us on the planet, so if we're not brought down by something during childhood or young adulthood, we will age. So I'll go over briefly some results for 5 tissues and organs, just to give you an idea that what you can do in one organ of the body or cell will also apply to others, or they may also be unique. Generally speaking, on senescent cells, there's a large industry at the moment where there are a lot of startups and a lot of clinical trials on the benefits of removing senescent cells. These are cells that not only do not do what they would have normally done, but they also secrete things that are deleterious to the cells around them. Fibrosis is a problem of aging. It's also a problem of repair after injury. You'd like to reduce it. And then some of the factors that influence this are proteins in the blood, which may be interesting in terms of development, further development to bring to the clinic, and also cells in the blood, which can be changed or which are there that already work. And then I'll summarize by saying what I just said. So let's just take a couple of examples from kidneys in 2 circumstances. If your kidneys are not functioning, your life will A, be miserable, and B, shorter. So from wherever the diagnosis takes place. So there's a class of compounds called senolytics. Dasatinib and quercetin are repurposed from chemotherapy and also from antioxidants. But they have the effect of reducing senescent cells. In this case, diabetes will result in kidney disease. And if you treat these diabetics, this is a 9-patient study from Jim Kirkland's lab at the Mayo Clinic, you will reduce senescent cells in the human body. So this is moving from the laboratory animal to the humans. You'll have these various markers of, well, it's too light anyway. So A, B, and C show you 3 different markers of senescent cells. Where it looks higher, that's without the treatment. Where it seems to be lower, that's following the treatment. So it's a first-in-kind demonstration for diabetic kidney disease that in a human you can reduce cellular senescence, or the burden of senescent cells. On the other hand, hydrogen sulfide, which we don't necessarily think of as a compound normally in the body. We didn't think of nitric oxide as a compound in the body either, but it's another gas that functions to signal, as it were, from one cell to another. Hydrogen sulfide can also ameliorate aging-associated changes in the kidney. Then the key is that there's reduced fibrosis. There's also reduced senescence from hydrogen sulfide treatment, improving the level, and the function of the kidneys is improved. So it's not just that you have some cellular changes or some molecular changes, you have an improvement in function. In the liver, an organ that under evolutionary pressure has evolved because the liver detoxifies all sorts of things that we come into contact in our environment. It's an organ where heterogeneity is a beneficial evolutionary outcome because you don't know what you're going to encounter next, neither does evolution, but the proteins expressed in the liver when the liver is functioning well help you to survive toxins. So here's something that, unlike Sean's Grubbly Farms, this is something done in the laboratory with fruit flies. Originally a factor found in fly brain turns out to have an effect in the liver. And also there's an example of combination therapy. Those of you who might apply to the NIH for a small business grant might think in terms of some of the things I'm presenting here. Everything I'm presenting, every scientist who's doing this either has their own company ongoing or a clinical trial in the works. But be that as it may, there's a lot of room to play in these fields. So this is an experiment where you join a young mouse to an old mouse. Something from the young mouse blood gets into the old mouse and it rejuvenates the old mouse. It can also go in the other direction. In this case, there's less fibrosis in the liver during regeneration. So you do a liver injury and something from the young circulation affects the liver regeneration. That thing from circulation turns out to be this protein called MANF. Well, that's a short talk. I have more slides. Is there some way I can get to them? Okay, so the next example is one from Irina Conboy's lab at Berkeley, where she used taking some blood from a young mouse and putting it into an old mouse so she doesn't stitch them together. It's a lot unpleasant for the young mice. And what happens there is that the liver function is improved. She's identified 2 compounds that work in this. One is an inhibitor of a thing called a protein kinase, and the other one is oxytocin. If anybody's delivered a child in this country in a major city hospital, you've been given Pitocin, which is a synthetic oxytocin. And it's also used in psychology. It has benefits in terms of people's moods. In terms of the liver, it helps in the regeneration, and it is optimized at a certain combination of the inhibitor plus the activator. So it also has an effect on brain function. It also has an effect in muscle regeneration and in other cells in the body. So these are proteins that are in circulation. The levels may change with age. The baseline level might be low. We don't really know at this point which thing is important. There are other proteins, for example, in muscle regeneration. So you have an injury and it repairs. There are proteins called clotho, which is in circulation. There's a protein called GDF11, also in circulation, and then oxytocin as well. These all affect muscle regeneration. So what is it to be illustrated here is that there are 3 ways in which you can get to the same regeneration problem, 3 different pathways. At some point, they have to converge, either at the level of the cells. Give me 1 minute. So, which means 2. So the key take-home from that one is that there are these molecules that affect multiple cells. Multiple molecules will also affect the same pathway. In muscle repair, there's also a feature of fat cells are in the muscle, and there are macrophages, immune cells that penetrate into the muscle, and they can the macrophages can affect a population of cells to either become more fat or more fibroblasts. The more fibroblasts, they induce fibrosis. So here you have a balancing situation between activation of the muscle repair pathway and activation of a fibrotic pathway. The older you are, the more likely it is for the fibrotic pathway to win under normal circumstances. That can be influenced. The things that influence all of these tissues also affect cognitive function. Macrophages in circulation can reach the blood-brain barrier. That information is translated across that barrier to reduce inflammation in the brain and improve cognitive function. My institute has $2 billion to spend on Alzheimer's disease, searching for cures, and trying to bring those cures to the clinic. So in summary, what I would tell you is that if you are thinking in terms of looking at aging as a treatable condition, there are multiple outcomes that can be looked at. The FDA does not have a single indication for aging, but there are aging-related diseases that are being targeted at the moment, and that there are multiple ways of getting to any of the conditions of aging. Thank you.